Good afternoon, and welcome to Simon Says. Today's session is on breast cancer and Black women, research to reduce disparities. I'm Lisa Hayes, and I'm the Executive Director of Pink Forever Ending Disparities, and I'm also the Director of Women's Health for Genesaret Free Clinic. At Pink Forever, our mission is to eliminate breast cancer disparities for Black women. So I was certainly honored when I was asked, and happy to be asked, to moderate today's session. And it's been my pleasure to work with some of the researchers that we have on today, as well as other IU researchers, not only because they're very knowledgeable and impactful people, but also from a personal perspective. As you can tell, I'm a Black woman, but I'm also a survivor of triple negative breast cancer. And so this groundbreaking research that these IU researchers are involved in have a personal connection for me. And I'm really excited today to hear what they will have to share. So I am going to go ahead and introduce our presenters for today, and then let them talk, because they certainly know more than I do, and I can't wait to hear all about what they're doing. So first of all, I want to introduce Dr. Kathy Miller. She is the Baobab Professor of Oncology and a leader in designing and leading clinical trials focused on breast cancer. Her work has implications in the treatment women receive every day. In fact, there are only a few drugs that are part of breast cancer care that Dr. Miller has not had at hand in testing. And our other presenter for today is Dr. Hari Nakshatri. Did I get it right? Nakshatri. And he is the Marion Morrison Chair of Breast Cancer Research, and as a leader in breast cancer, normal or healthy tissue research. He's upending previous assumptions about how breast cancer develops and the unique ways it affects women from different racial and ethnic backgrounds. So with these introductions, I am happy to introduce both of them to you. And I think we're going to start off first with Dr. Hari.
Thanks, Lisa. Let me share my screen. And I'm going to start off with a brief presentation about the work that happens in the lab setting and how that research is helping us to understand the breast cancer disparity. So for many, many years, a lot of us researchers are focused on studying what happens in the earliest stage of breast cancer, ductal carcinoma in situ. Then we changed our focus over invasive carcinoma. So then we started looking at why the breast cancer spreads to the different parts of the body. At the same time, we completely ignored how exactly the normal breast looks like. Yes, there has been a research going on with the mouse model, where we call mammary gland. The structure of the mouse mammary gland, while useful to study, it is completely different than what is in human. So the idea then is we should be studying the normal breast of healthy women before we can understand what genetic changes that happen in the ductal carcinoma in situ, invasive carcinoma, and metastatic cancer. Then an additional layer of complexity came in once the genomes, we started sequencing the genome. To what extent the normal is similar between the individuals. So that is the next focus we need to look into. So what my lab and we at IU School of Medicine and Simon Cancer Center is looking into, to what extent are the normal breast cells are similar between individuals? Are they similar across our different genetic ancestry? The meaning of genetic ancestry is your ancestors either from Europe, Africa, or the Latina and Asia. Are they similar? Are they dissimilar? If they are not similar, to what extent that plays a role in the health disparity that we are looking at? Most of the health disparity research was focused on the socioeconomic and other factors. Can biology play a role as well in this health disparity? To understand those complex questions, first we need to study the normal breast itself. So the kind of questions we started asking over the years is, how many different cell types are present in the normal breast? That's the number one question. The next question, once we address that, is, does the cell composition differ between individuals depending on their genetic ancestry? Then we started asking question, there are BRCA1 and BRCA2 carriers who are highly susceptible for breast cancer. Do their normal breast cells differ from those who do not carry those risk elements? So the next question we asked is there any specific cell type? If, for example, if you consider there are 100 cell types or 100 building blocks in the normal breast, which among them are more susceptible to become cancerous than the ones which are not? So then we can start focusing our biology work on those cells. The last question, which we have not started asking yet, but we hope to ask in future, what effects do age, pregnancy, lactation, and then evolution have on the cell types of the breast? This is becoming critical because we do know that the breastfeeding has an impact on the type of breast cancer as well as how it prevents certain types of breast cancer. We do not know the biology behind that. Let me just give you the snapshot of the first question we addressed. This is a kind of study we recently published where we showed that there are at least 23 different types of cells in the normal breast. That's the number one finding we made. This is using the normal tissue many women, some of you are in the audience, donated to us for only research purpose. Without your contribution of this tissue, we would not have achieved this feat. And this has now become the part of the global efforts to develop the entire body atlas. And we are very happy, pleased to see that Simon Cancer Center contributed to this. And all this data are shared through this website to the entire world. What we found was that there are 3 cell types here and 1 cell type here from which the majority of the breast cancers originate. So knowing that, now we can come up with a model system to figure out how the cancer originates and whether these 4 cell types that we identified, do they vary based on your susceptibility to breast cancer or not? So that is the next step we are pursuing. And this is a very complicated assay. Per sample, it costs about $10,000 to do this, but we want to come up with a cheaper assay so that we can apply this in a routine basis. The next question we addressed is with the BRCA1 and BRCA2 patients. What we found, this is again a similar assay. What we found, these are the common cell types present in both the normal and BRCA1. And this one cell type, one type of cell, is missing in BRCA1 patients. So maybe this one type of cell, which helps the other cells to be under normal condition, prevent them from undergoing rogue, but that control cells are missing in BRCA1. So we are trying to figure out how these cells are helping. Along the line, what we found is that if you take these cells from healthy donor, we can make them cancerous in the lab. But you need to have 3 to 4 type of mutations you need to introduce them and to make them cancerous. But if we make the same cells, get the same cells from BRCA1 and BRCA2 patients, carrying the BRCA1 or BRCA2 mutations, we can make them cancerous with only 2 genetic aberrations. So that way we have now been able to show or figure out how the BRCA1 and BRCA2 mutations predispose for breast cancer. The next question using the same common normal tissue we started addressing is, are there any genetic ancestry dependent differences? Now, this is a type of cells. This is the normal way inside the breast the cells look like. Most of the breast cancers originate from them. And there is a other cell type which surround them so that they can keep them healthy. And we found this unique cell type which looks brown, what we call a marker for ZEB1. These cells which surround this duct are higher in the normal breast of women of African ancestry. And these cells increase in the breast of women of European ancestry only in the breast that has cancer. So what role these cells are playing? That is something we are looking at, why they are high. If they are high in African American women, how do they contribute to the breast cancer? So in this contrast, what we are working at the moment, we have some evidence, is to compare the breast cancer to a different type of trees, for example. You can see the three types of tree here: palm tree, chestnut tree, and baobab tree. Palm tree, if you want to cut it or get it out of the ground, it's very simple. It doesn't have too many roots and too many branches. It's easy to get rid of. It's like a surgery removing the breast tumor. Chestnut is a bit more complicated. There's more branches, the roots are a little bit deeper. You may need not only surgery, chemotherapy. By contrast, baobab tree, it's pretty much impossible to get rid of. That may be the most aggressive cancer types. So what we are trying to figure out that the unique cells that are enriched in African-American women, will they make the tumors in them much more like a baobab tree? Or will they make them a chestnut tree compared to the palm tree? So that is the kind of studies that we are doing, and those work are going on. So as a researcher, we always have to have a long vision, right? So one of the areas that with the common tissue that we have, we want to focus is what we call is what I call is oncologic anthropology. Many years ago, we lived in different parts of the world. We got adapted to marine diet. We got adapted, and the milk was not, came into this evolutionary milk is not for human consumption. That was for the cows to rear their calves, and then that's, but we started using them. Now we adapted to lactose. Some of us still have the lactose intolerance. And certain areas, malaria is pretty endemic. So the genes in us got mutated in such a way that we can adapt and resist the malarial infection. Same thing with the cholera. Same thing is arsenic-rich environment. And if you look into certain part of Bangladesh or Western Bengal, the rice you grow there has ton of arsenic because the soil is full of arsenic. But somehow we underwent mutations to adapt to that. Same thing with the cold climate, altitude, light skin, and short stature. We adapted to this for many years ago by mutating certain genes. And here is an example of a gene that is mutated in people in Tibetan plateau, where the oxygen levels are so low that our genome had to change to adapt to that oxygen climate. But now we are very motile. People who have these genes which help them to resist malaria, they're in the United States. And people who are the Tibetan people in Tibet, now they are in Bombay at the sea level. Or some of us who are who grew up in New York, now they are in high up in the Colorado mountains. But we have not adapted to those changes. If you carried those genomic changes that helped your ancestors to adapt to a different region, now do you move to a different location? If they develop cancer, will those adaptive changes influence the way the cancer behaves? The reason I'm bringing that up is that we have taken a gene called Duffy. A Duffy null phenotype is more common in women and the people in sub-Saharan Africa that confer them resistant to malaria. But if some Duffy carrier gets breast cancer tend to be incredibly aggressive in them. And we have developed the models that shows why they are aggressive. So these are the what we call adaptive traits we need to adopt in our breast cancer research to figure out whether somebody with this adaptive trait, will they respond differently to a chemotherapy or any targeted therapy compared to the one who do not have this adaptive trait, although they may be of the same genetic ancestry. So moving forward, what we think is we need to start up with a kind of an oncologic anthropology. This was initially coined by Dr. Lisa Newman. Her thought was social scientists and geneticists and translational oncologists should work together, bringing these adaptive traits. In my lab, what I would like to do is I would like to use the common normal tissue and figure out whether any one of them carry these adaptive traits, which I talked to you about. And if I make those cells cancerous, will they behave differently compared to the cells that do not have those adaptive traits? As I told you, we have already worked on Duffy, and which seems to be working in that direction. So this is my broader vision on how the research will go using our common normal tissue. So I'll stop there and take the questions later on after Kathy's presentation. I'll stop sharing my screen.
Thank you, Dr. Hari. Go ahead, Dr. Miller.
So I did not prepare slides because what I want to talk to you about is a little bit more simple than the really elegant, detailed, basic science work. I spend a lot of my time thinking about the things that Hari is uncovering in biology and wondering how could that impact the care of patients in the clinic, which is where I spend the majority of my time. So if we think about the impact of Hari's work, it has tremendous potential impact across everything we do. So for years, we have known that Black women have a higher mortality from breast cancer. And we've assumed that that's because of access to care and socioeconomic disparities. But when we've looked at the outcome of women who were treated on national clinical trials, so they had access to care, they had access to those trials, they were treated in the same way, Black women still have a worse outcome. And Hari's data gives us a potential biologic reason for why that might be. It may simply mean that the disease they develop is more aggressive because there are differences in their breasts even before they developed breast cancer. And if that's the case, that could mean we may need slightly different treatments for women of African descent. It may mean in the future that we need to think about different screening methods. It may mean we need to think about different preventative methods. But there are other ways that those inherited genetic differences could impact outcome. It may impact how our bodies metabolize drugs or how sensitive or not sensitive we are to particular toxicities that can occur with our chemotherapies. So one of our colleagues, Dr. Brian Schneider, is particularly interested in how drugs interact with our bodies and how that is individual and how our genetic makeup may impact how drugs interact with our bodies. Brian used data from two very large national clinical trials that treated patients with early stage breast cancer with chemotherapy. And what he found is that African-American women had a worse outcome. And when he looked at this common side effect called chemotherapy-induced peripheral neuropathy, numbness, tingling in the fingers and toes, then when it's mild can be just some annoying numbness, but as it gets worse, can be painful and can impact the function. And being able to do fine movements with your hands, feeling wobbly or unsteady on your feet because you can't feel where you put your feet when you're walking. That side effect was much more common in African-American women, and it was much more severe. And in particular, with the commonly used chemotherapy medicine called Taxol, Brian found that African-American women were more likely to have lower dose reductions of their chemotherapy driven by this increase in neuropathy, and they had a worse outcome. He's identified some specific genes that are inherited much more frequently in African-American women that seem to explain that difference in the risk of that particular side effect. And really importantly, he looked at a related chemotherapy medicine called Taxotere. Taxotere can also cause peripheral neuropathy, but with Taxotere, we didn't see a difference in the likelihood of neuropathy or the severity of neuropathy in African-American women. There wasn't a difference in dose reductions, and there wasn't a difference in outcome. Now, Taxol and Taxotere are not the same drugs, but they're very similar. And when their effectiveness has been compared head to head in a large group of women, most of them Caucasian, there's no difference in their effectiveness. And that really raised the question that there's no difference in effectiveness in Caucasian women, but maybe there is in African-American women because we have more side effects with one and you can't get the full treatment. But that's not the case with the other drug. Maybe that's the drug that you should use preferentially. So Brian and his colleagues are currently leading the first, and at this point still the only clinical trial specifically for African-American women with breast cancer, who because of the type of breast cancer they have, are going to receive treatment that includes either Taxol or Taxotere. It's not randomized. Those two drugs are used differently based on when they were developed and the regimens that were developed. But their standard treatment is going to use one of those two drugs. Carefully follow them for how much neuropathy they develop, whether they had dose reductions or shortened the course of chemotherapy. And we also get a genetic sample to look and see if they've inherited any of those genes that Brian identified that marks for an increased risk. Our hope and the goal of this study is if it confirms the early data, that will lead to a recommendation African-American women be preferentially treated with Taxotere instead of Taxol, because it reduces the risk of side effects, allows them to get a more full course of therapy, and because of that, gives them more effective treatment. Now, imagine if African-American women had not participated in those two large clinical trials that Brian did. We would have never known that this difference existed to be able to study this further. And it was only possible because those clinical trials each enrolled around 5,000 women. So even though the proportion of African-American women in those trials were fairly small, we were able to see that difference and pursue it. There's been a lot of thought about why African-American women and men are less likely to enroll in clinical trials. And there are certainly many reasons, but a big one is they're less likely to be asked to enroll in clinical trials. And what we have learned from this trial limited to patients with African-American women is if you ask them, if you design a trial that specifically addresses a question that is important to them and will impact them and their community, they are more than willing to participate. Enrollment to this trial before the pandemic derailed lots of things. Enrollment to that trial was actually ahead of the pace that we had predicted and that we had set out. This is a really compelling story, that the richness of humanity has implications in ways that we had only imagined. And if we can ask those questions, collect the biologic samples, whether they be biopsies or blood samples, and begin to interrogate those differences, the understanding that that will bring to us and the ways that that will impact treatment are really nothing short of amazing, and makes this such an incredibly rich and fascinating time to be part of this field in medicine. So Lisa, thank you for the work you do in helping us all do this work. This is a simple philosophy, perhaps best said, no work about us without us. So we really need to continue to focus on being partners with our patients and making sure that the questions we're asking are important to them. And when that's the case, that partnership allows us to take the work even farther.
Thanks so much, Dr. Miller. So much great information shared already, and we have some questions that have been submitted today and some even earlier. But before we get to those questions, I did want to bring up something that in my experience in working with survivors and people currently in treatment, one of the challenges they have is the timing of when the information is presented to them about possibly enrolling in a clinical trial, because that typically when you're diagnosed, you go for that first consultation and you were just overwhelmed with information, and so much so that it might be too much to even think about a clinical trial, what that means, and whether or not you should participate. So do either one of you have any thoughts about how to make that timing work for the patient?
So I'll tackle that one, since I spend more of my time in clinic than Hari does. When you're first told that you have cancer, many patients tell me the world kind of goes blank. for a while, and suddenly they realized, oh, that person was still talking. I don't really know what they were saying. I often explain it to my patients is, I become like the adults in the Charlie Brown cartoon, where all you're hearing is wah, wah, wah. I think it's important to remind patients early and often that this is not an emergency. It is urgent, and not that we want to delay things, but this is not an emergency. There's time to take a breath, let that immediate panic response calm down a little bit, get other opinions and get the information that you need to understand your disease, what's being recommended to you and what's being asked of you, and to make certain that you're comfortable that this is the right plan for you and this is the right team to be working with you. I think it's also helpful. I will call this normalizing clinical trials. I think often people have the idea, particularly in cancer care, that clinical trials are what you do when there's nothing left to do. You don't think about clinical trials until you're at that very late stage of disease. And there certainly are clinical trials for patients at the very late stage of disease. But there are also clinical trials asking questions that are important for women just diagnosed with their breast cancer. There are clinical trials looking at different methods of screening, clinical trials looking at preventative strategies. Anywhere that we have questions about how to best tackle this problem in the clinic, there are or there will be clinical trials. So we try to spend a lot of time talking about clinical trials early, talking about clinical trials in the community, to demystify this process and make it seem less scary, make it seem like something we should talk about anytime we need to make a change in treatment. Anytime we need to intervene, one of the questions should be, is there a clinical trial that it is appropriate for my situation? Is that something I should be considering?
So making it part of the normal discussion during an encounter with the provider.
It is part of what we do. We are trying to launch a new initiative, Lisa, that's very simple. That includes when people first call and schedule an appointment with us, in addition to telling them details of where to come and when to come and what documents to bring and when to check in, to also tell them research cures cancer. And we do clinical trials here. So as part of your visit to our center, you may be asked to participate in clinical trials. Not everyone will be asked. There is not a clinical trial that is appropriate for every single patient we see, but we would like everybody who comes to us to know that we are very proud of the research we do. We think it's really important because research is what cures cancer. And you have the potential to be part of that.
Exactly. That's a great way of giving back as well. And, one of the takeaways from many things you have said for me is that the decision doesn't have to be rushed. The provider doesn't have to rush, nor does the patient have to rush. Take some time to get all the information and make informed decisions. So that is great. I'm going to move on to one of the questions submitted earlier during your presentation. The question is, so much is being learned with the resources we have at hand. What additional resources, equipment, sample sets, or public participation is needed to ensure that this research meets important goals in a timely manner in order to get them from the bench to the clinic?
Hari, this person, it could be my new best friend. I'll let you tackle what you want on the basic side.
Yes, I mean, at the moment, as I mentioned, most of the assays that I presented are in an expensive assay. We do it at one sample at a time or 5 samples at a time. Now, what we want to do is how can we apply this to say, for example, you get a piece of slide with a small piece of tissue attached to it, like the way pathologists get it. Can we do these sophisticated studies that we have done so far using that little material and in a little time? The data that I presented today is about 3 to 5 years of work. but I just gave it in one slide or one picture, right? So we want to get it to a stage where we should develop additional assays to such a way that the clinicians can use those data in real time rather than waiting for a couple of months to make to take a treatment decision. So that is our future goal. And again, the next thing what we would like to do is we would like to study the individual tumor of a person rather than clubbing them into ER positive, ER negative, one has a TNBC, one has a HER2 positive. No, we need to figure out a way to study everyone's tumor in real time and then help the clinicians to make a treatment decision. So that's our future goal.
So
Thanks,
I'll tell you what I need, Lisa.
Yes.
Currently, only about 3% of adults with cancer participate in clinical trials. And close to half of the clinical trials that start never get finished because there are not enough patients who enroll in those trials. That is such tremendous missed opportunity. Imagine how much faster we could ask and answer those questions if twice as many or 3 times as many or 4 times as many of adults with cancer were treated in clinical trials. To give you an idea of how poorly we are doing, over 80% of children diagnosed with cancer are treated on clinical trials, but only 3 to 5% of adults. The other thing we need, honestly, is an investment of money to support that clinical research and to support the basic research. So when I last looked at this data a few years ago, we as a country spend more on soft drinks and cat food than we do on cancer research. If you want all of the sources of research, government, pharmaceutical companies, private foundations, lump them all together, we as a country spend more on soft drink and cat food. The tobacco industry spend more on advertising than the National Cancer Institute is able to spend on cancer research. So if you imagine the clinical trials that were started with a great idea, and we were never able to answer that question because it didn't get done. So that's why we're so passionate about the research. That is how we move things forward. Everything that I'm able to tell my patients, this is the best currently available treatment for you, at some point was the new idea in a clinical trial. That's how we learned this is the best current trial for you. So unless we're satisfied that we're doing as good as we need to do right now, the research needs to continue. And that takes that commitment and evaluating our priorities and where we want to put our resources and our time.
Yeah, so adult participants, but the statistic that you shared is kind of startling to me that more than 80% of children will participate, but not those adults. But obviously those are adults that are consenting on behalf of their child. It might be kind of speculative, but why, if an adult is willing to let their child participate, why does that translate to an adult participating in a clinical trial?
So it's an interesting question, and actually it would be fascinating to study the family members of children who had been impacted with cancer to see if those family members were more likely to participate in a clinical trial. As adults, they had that opportunity compared to adults who did not know a child in their family affected. I think part of it is where those children are treated. Most children are treated in larger academic centers or larger community referral centers that actively participate in research. And most adults are treated in community practices, many of which don't actively participate in research. So I think that's part of the difference. I'm sure there were other differences as well, but I like the way you framed the question, Lisa, because I'm not sure anyone
Thank you. I'm going to move on to some questions submitted earlier. The first one is, breast cancer disparities in the Black community have been an issue for quite some time. in your opinion, is the most pertinent reason we seem to struggle with finding adequate solutions to this issue?
I'll take the first part. I'll take the first part on this one, Lisa.
Okay.
Not only do very few adults participate in clinical trials, but the participation for communities of color and minority populations tends to be lower. That certainly is not always the case. But if we are not able to engage those communities and study those communities and questions that are important to them, then there will continue to be that gap. Part of it is in the way we design clinical trials, quite honestly. Clinical trials often require extra visits to the clinic, then they require extra procedures. That's additional transportation, it's additional time away from work. So if you have a population that has less access to resources, that has been historically disadvantaged, those logistics can make this more difficult. It doesn't make it less important, but we need to recognize those things and have the support there to make it possible. We also have designed some of the eligibility criteria for the clinical trials to be so strict that some of our minority patients are less likely to be eligible. So exclusions for other health problems, exclusions based on diabetes, based on obesity or high blood pressure, more likely to exclude patients of color than Caucasian patients.
Right.
Simple things like we know many African-American women tend to have slightly lower blood counts. Their blood counts are normal for them. They are perfectly healthy. But that shift in a normal range of blood counts for an African-American population means some African-American women will be excluded for something that is normal. So there has also been a more recent focus on looking at the impact of those technical aspects of the clinical trial and how they might inadvertently exclude people. While at the same time, we're trying to include them.
Right. Did you have anything to add, Dr. Hari?
Yeah, I mean, one of the reasons why is that from the biology point of view, for a long time, people didn't think that the biology plays any role. But once we started getting the sequence information and clinically this targeted therapy started showing with the same mutation, for example, in lung cancer, one of the common mutation is a gene called EGFR. If an Asian with the EGFR mutation has lung cancer, they respond better than a person of European ancestry. Those kind of data started coming on, then people started to think that there must be something in biology as well. Now, and then of course, understanding came about, but we didn't have any model system. How do we study that? With this tissue bank that we have, now we are in the process of developing the model system to study it. I hope within the next 5 to 10 years, we can start addressing the biology questions and take it into clinic.
That would be incredible.
Yeah.
So kind of related to that, I mean, when you describe your research in Dr. Snyder's research, clearly cancer treatment is moving more toward targeted therapies and precision health. So if minorities are not included in this research, and that's the trend now of going to those kinds of therapies, what does that mean for those populations?
So it has the potential, Lisa, that those disparities could grow instead of narrowing. So if the ability to precisely target treatment to something specific in your tumor is effective, but minority populations are less likely to be treated in centers that have that sequencing technology available. We don't understand the different genetics that often arise in their tumors, or the impact of those genetic differences. Then unfortunately, it may mean that those disparities grow rather than getting narrow. But it has the great potential to narrow those differences if we're able to engage with those populations and ask those questions. And I think Brian's study focused on neuropathy is a great example.
Right.
Using that precision medicine approach, he identified a difference based on genetic ancestry. He's identified genes that biologically seem to explain that ancestry or that difference. And in work that I didn't talk about in detail in his laboratory, he's trying to understand the function of those genes. How is it that those genes impact this risk of neuropathy? Because that could also lead to ways of preventing the neuropathy or treating it, so that women who really need those drugs may be able to get the full benefit of that. But that work has the potential to narrow that difference and narrow those disparities by changing the treatment of a minority population based on what we've learned. But none of those things are going to be possible if we can't get the patients and the samples and the communities behind this approach. Unfortunately, in today's world, it can be very difficult to talk about racial differences without that sounding negative and that we're trying to drive people apart. When the goal is simply recognizing that genetic inheritance matters, impacts our biology, and we need to take that into account so that we can really best treat this whole person. And that means knowing who she is, what's important to her, where she came from, how her body might react to drugs, good and bad, so that we can take all those things into account.
Right. I think a different way to look at it rather than differences is inclusion rather than exclusion. That's a better way. Kind of related to what you were just talking about, Dr. Miller, but I'm going to address this to Dr. Hari. Someone asked this question, said, I would like to hear more about how the breast tissue bank is doing with regards to Black women donors and how the information has been used so far. And I know you've touched on that some, but can you expound on that a little more?
Right. Again, one of the cells that I showed in my presentation that we were able to identify only because we had enough tissue from Black women and we could compare it with the women, the European ancestry women and Latina. So that's the only way we could figure out that is a fundamental difference in the normal biology. That then extended to the work on the Duffy phenotype. 40% of women who donated the tissue had a Duffy, partially Duffy or fully Duffy phenotype. So that is really helping us to characterize the other part. And in that adaptive trait, what I showed you, there are many, many other things which seems to be different in people of African ancestry. So we can now start taking one after the other and try to define that even further in a finer manner. So this tissue is an enormous resource for us, not only from understanding the basic biology, but I think eventually it will be translated into clinic.
Great.
Lisa, can I add? I know you know this because you've helped us do this. But the work of the tissue bank to reach out to those populations has been very intentional. Talking with leaders in those communities, trying to develop very culturally grounded communication materials and recruitment materials, and going on the road. Back in the days when people actually traveled,
Right.
having tissue collection events in Texas to recruit a Latina population, in Detroit to recruit an African-American population, in California to try to recruit a bigger Asian population. Our distinctions right now are at a really gross level. I strongly suspect there will be significant differences if we talk about women of African descent. Are we talking about Northern Africa or Sub-Saharan Africa? There will be regional differences within those gross differentiators. I can only hear my Japanese and Chinese and Indian colleagues cringing at the idea of lumping them all together as Asian, as if those differences didn't matter. We're not at the level of being able to get to that discrimination yet. But we will be, and we will need to be.
So again, it goes back to needing a more diverse group of participants, whether it's for the Komen tissue bank donation or for clinical trials. It's all about
Absolutely.
Inclusion again. I think we have time maybe for one more question. And so I don't think we can have Simon Says without bringing up the pandemic at all, because I'm sure it's had an impact, but this is also a question someone submitted. Are you seeing an increase or decrease in the number of breast cancer patients in Indiana over the past couple of years? And has the pandemic attributed to the amount of care available to patients?
So it definitely has. I don't think we have the numbers yet from the state on the number of women diagnosed. We have definitely seen decreases in the number of women coming for mammography screening. And honestly, in the very early days of the pandemic, we discouraged people from coming to hospital before there was a vaccine, when the virus rates were very high, for routine screening or elective procedures. That risk and benefit did not make sense. But unfortunately, that means there are a lot of women who had been getting annual mammogram screenings who didn't get a mammogram in late 2019 or anywhere in 2020. I've seen several patients recently diagnosed now in 2021 who had a gap in screening driven by the pandemic. These are very responsible women who took good care of themselves, but that gets very frustrating. We certainly saw periods of time where we needed to delay breast cancer surgery because of the impact of the pandemic, just on the ability to have healthcare services and the resources available, with so many of those resources needing to be devoted to people with the virus and active infections. That is less of an issue now, but we're still not able to schedule elective procedures, elective reconstructions for patients in the same sort of time frame. So it has forced us to think a lot about prioritizing care. How can we make sure that women get care that they need for their breast cancer, but in a way that is safe and responsible, without exposing them to risk of the pandemic? Strongly encouraging people to get vaccinated now that that's available, because right now our biggest limitation is the resources. And all of the resources still have to be devoted to taking care of people with the virus, the majority of whom need hospitalized care having not been vaccinated, still impacts our ability to care for patients with cancer.
Well, thank you so much for sharing, Dr. Miller, Dr. Hari. The information has been incredible, and I know that the audience that tuned in has been enlightened, and I certainly have too. So I think we're about to wrap up, but I'll leave with this, that hopefully soon there will be another Komen Tissue Bank donation event. So after all you've heard about the importance of having tissue donations from a diversity of women, please consider donating, or if you are a cancer patient, please consider participating in a clinical trial. If you have not been asked, be proactive, ask yourself, is there something that I can help with in a clinical trial, or something possibly that can help me? So thank you again for everyone for joining us. Thank you, Dr. Miller and Dr. Hari, for sharing with us today.
Thank you.
Thank you, Lisa.
Thank you all.